So again, I also thank you for the audience. It's really a pleasure to meet your audience, and given that it's evening in Algiers. So as Abdelghani said, I believe many of you are familiar with recent development in machine learning, deep learning, including ChatGPT and DALI. The emphasis of my talk today is somehow different. It's really about how can we use deep learning, machine learning to tackle important scientific question in biology and drug discovery in particular. So I will try to give you sort of a glimpse about AlphaFold 2, this system by Google DeepMind, our own work, OpenFold, and stress kind of the sort of perspective that they are opening for research, but also for industry and drug discovery. So because I believe this is a mixed audience, you have many different backgrounds. Before diving into machine learning, let me tell you a little bit about this problem. What is the scientific problem that was, to a large extent, solved by AlphaFold 2 and OpenFold, and why this problem is important? So the problem statement is protein structure prediction. And unlike many other problems in biology, the statement is very simple to understand, including for computer scientists. So about half a century ago, biologists realized that much of the information that is needed to fold a protein and achieve the three-dimensional structure that you see here on the right is included in a one-dimensional string of amino acids or discrete tokens if you are a computer scientist. So since then, it has been a major challenge to write down a computer algorithm where you feed in a sequence of amino acids, an alphabet for biology. There are 20 types of amino acids. And the aim of your algorithm is to predict with very high accuracy a three-dimensional protein structure. So I hope you realize that from a high-level perspective, the statement of the problem is quite easy to understand. Give me your sequence as an input and return highly accurate structure. I should maybe add few words why this problem is very important in biology and to some extent drug discovery as well. I believe you are all familiar with DNA. DNA is sort of the hero of 20th century biology. We think about the molecule, the double helix, but really the most important molecule in life are proteins. If you think about any function that is happening in your body or any organism, in many cases, those functions are encoded into proteins. And the way how those proteins achieve their function is to a large extent encoded into their three-dimensional structure. So basically, understanding life at the most fundamental level starts with those three-dimensional structures. Many diseases are also connected to situations where proteins, they don't have the right shape. So many diseases, including neurodegenerative diseases, are situations where some proteins are not doing the task that they are supposed to achieve. And this is encoded in kind of their structure and what we call misfolding. And so this question is extremely important. It has been a challenge for half a century. The only way to get those structures until very recently was to use experiments. The problem is experiments are tedious, they are very expensive. To characterize one protein structure, typically you need months, if not years. You need to spend thousands, if not hundreds of thousands of dollars, and have very sophisticated teams working on that problem. So today, with the AlphaFold 2 breakthrough that I will explain in a moment, we are able to fold those structures, in many cases, in seconds, using the deep learning and machine learning kind of development. So this is really a remarkable advance. And from my perspective, this is the most important application of deep learning, machine learning, not only in biology, but in any scientific problem beyond biology. And that's kind of what I would try to share with you today. What was the situation a few years ago? Where are we today? And what kind of machine learning techniques we are using to tackle this problem and problems beyond protein structure prediction. I will be also talking about our own system, OpenFold, and this is a kind of quote from Yann LeCun. I believe many of you in computer science are familiar with this name. He's one of kind of the leading person in machine learning and deep learning. And I just read Yann LeCun quote: OpenFold is an open source, trainable, memory efficient, GPU-friendly PyTorch reproduction of AlphaFold 2. And I will unpack those words later in my talk and really kind of stress the idea of like being trainable. I will explain later that AlphaFold 2 code is available. People can use it for inference, but we cannot train the system because there are important modules lacking in the publicly available version of AlphaFold 2, whereas our system OpenFold is trainable. And the impact of being trainable is very important, both from a computer science perspective, but also from a kind of application perspective for questions in biology and drug discovery. So again, before diving into the machine learning part, let me give you sort of a brief historical detour to really understand the extent to which the AlphaFold 2 moment was a turning point in biology and structural biology. So as you know, in machine learning, benchmarking is very important, and the same for structural biology and biology. We do have benchmarks to assess the quality of our computational systems. And moreover, in structural biology, we have a sort of protein Olympics. It's an experiment, or you might call it a competition, happening every 2 years, where the organizers assess the best computational system for protein structure prediction. And this is really, for us, the gold standard for the following reason. As you may know, in many cases in machine learning, deep learning, you can fool yourself thinking that you are achieving very high accuracy, but you are not benchmarking your system in an appropriate way. In a nutshell, what this experiment is about, the organizer of the CASP competition or experiment, they will release the sequences that I mentioned earlier, the one-dimensional sequence of tokens for certain proteins. The structures are unknown, and the participants have like few days, weeks to return their prediction. So the remarkable thing about CASP is that the predictions are done on a blind fashion. You don't have access to the experimental structure. You don't have access to the ground truth, as we say in machine learning. Therefore, you cannot fool yourself thinking that you have a very highly accurate system. So let me kind of go back here about 10 years ago and explain what was the situation. So the metric that you see on the y-axis, RMSD, is the metric that assesses the quality of your prediction. The lower, the better. 0 means a perfect prediction. It means that your computational system is predicting proteins with perfect accuracy. So from 2012 around 2016, the very best systems submitted at CASP, this again experiment or competition, they were above 10 angstrom. Details are not important. What is important to notice is that those predictions, in a way, they are not better than just having a noisy structure. So basically, 10 years ago, we were struggling. We were not able to write down computational algorithms that could provide accurate protein structure prediction. A turning point happened in 2018 where DeepMind and Google, they entered this competition with their first system called AlphaFold 1, and they did much better than anyone else previously. And as you see here, according to this metric, they did substantially better than any system prior to AlphaFold 1. That was a major development, but despite the development, that was not yet of interest to experimental biologists. Having a prediction that is above 5 RMSD angstrom is not yet allowing you to build experimental hypotheses. So 2018, AlphaFold 1, major achievement from a computational perspective, but it was not yet impacting experimental biology and biologists more broadly. The tipping point, the phase transition, happened at CASP 14 in 2020, where the system AlphaFold 2 was able to predict protein structures with an average of 2 RMSD. And that was a turning point. This is the time when we entered the regime where those computational pipelines and deep learning systems are achieving an accuracy that is almost as good as what you can get in an experimental lab. And since then, AlphaFold 2 and our OpenFold, they became sort of a toolkit that is used by structural biologists and biologists more broadly. So that was, again, the turning point. And the purpose, again, of this plot was really to give you a sense how things were developing very fast in the last couple of years. So let me just give you an example. These are what we call a cartoon representation of proteins. You don't need to be expert, but you will realize there are alpha helices and beta sheets. And again, you don't need to be an expert to recognize that the experimental results here plotted in green are almost perfectly matching the computational results predicted by AlphaFold 2 in blue. And that was the turning point. What you see again in green, this is a structure that was characterized by experimentalists in a lab. And what you see in blue, this is the prediction of AlphaFold 2. And that was, again, the turning point in our space. So one thing about those predictions to keep in mind, which is very important, if you are familiar with systems like ChatGPT, a major conundrum are hallucination. I believe many of you played with ChatGPT. It is a remarkable system. You ask questions many times. We are really surprised by the system abilities, but also we recognize that the system is also hallucinating. And it's a big research question. How can we distinguish between situations where systems like ChatGPT are providing good insight and situations where they are hallucinating? You can ask the same question for proteins and protein structure prediction. How can we really assess the quality of the predicted structure if we don't have a ground truth structure, if we don't have experimental structure? And so basically, how are we taming the hallucination? It turns out that something extremely remarkable about AlphaFold 2 and our OpenFold, unlike ChatGPT and unlike all the kind of familiar machine learning system, the predictions are calibrated. So what do I mean by that? On top of predicting a structure, we assign a local confidence score that tell us whether the machine learning, deep learning pipeline is confident in its prediction. And this is a remarkable tool for experimental biology. So let me briefly unpack this. So this is an example of a protein predicted by AlphaFold 2. You see that there is a color coding scheme. In a nutshell, the dark blue region are predicted with high confidence. It means that AlphaFold 2 is very confident that those dark blue regions are correct. The orange, yellow region are predicted with low confidence, which means that the system itself recognizes that those regions might not be predicted correctly. The AlphaFold 2 team did a very thorough assessment about those calibration. We did it on our end, and the very strong evidence today is suggesting that those confidence scores are highly reliable. And this is extremely important because if you're an experimental biologist and you turn the algorithm and you make a prediction, you can now know whether you should take very seriously the prediction that is coming from AlphaFold 2 because you have this confidence score. So one immediate impact of AlphaFold 2 is that we are starting to do science, or at least part of science in structural biology, at digital speed. So I mentioned earlier, we can characterize proteins using experimental techniques like X-ray crystallography that might be familiar to physicists in the audience, but those techniques are tedious, they are expensive, and this is the reason why we started using those techniques in the late 50s and early 60s from the previous century. And until now, we have only characterized over 200,000 proteins. So that was the situation. If you look at databases for structural biologists, the main database is called Protein Data Bank. We have about 200,000 structures, and there is no hope to kind of substantially scale those structures using experiments because the experiments are tedious. So what happened with AlphaFold 2 and OpenFold in our system is that the AlphaFold 2 team first, they released 1 million structure. That was in 2021. And then later, they have released 200 million structures. And again, you don't need to be an expert to realize the extent by which we were able to move from 200,000 structures to 200 million structures in few months using the power of deep learning and machine learning. And again, that is a remarkable achievement. And you can ask the question, what is the validity of those structures? We have 200 million structures, how good they are. The answer is the calibration score that I mentioned earlier and the confidence score. Because we have this highly calibrated output, we do know that about a third of those 200 million structures are predicted with very, very high accuracy. Therefore, they can guide biologists into their hypothesis and experimental work. So Now, let me just step back a moment and say why this is a very important discovery, not only for science, but opening prospects for drug discovery. Um, some of you are, I believe all of you are familiar with the Moore's law, which is capturing the fact that computing power is becoming faster and less expensive over the year. This is the sort of pink line or red line, depending how you see it on your end. Unfortunately, in drug discovery, we have a sort of inverse Moore law called Arum law by certain expert, which is the blue curve. Drug discovery is becoming slower and more expensive over time. And that's why some drugs are extremely expensive, and countries that are not in the West don't have access to those drugs. And this is also why we are not curing, or we don't have systematic strategies for curing certain diseases like cancer and neurodegenerative disease. So the question really is, how can we turn the blue line or the blue curve into something that starts looking like the Moore's law? And what AlphaFold 2 achieved by itself will not turn and flip the coin for the blue line, but it could be the beginning of a situation where AI, because it's fast, efficient, and accurate, it will substantially allow us to increase our abilities to bring new drugs to the market with reasonable prices. So that was, again, giving you a sort of a taste, a glimpse about what was the problem about protein structure prediction, why this problem is important, and what was achieved in terms of metrics and in terms of impact by folding millions of proteins. However, and this is connected to the drug discovery problem, proteins by themselves, they don't do much in a living organism. To really understand the function of the protein, you need to understand the structure, as I stressed earlier, but you really need to understand how proteins form complexes with other proteins. So this is an example where you see protein binding a different protein, how proteins bind with DNA or RNA, and how they bind with small molecules. For example, typically the drugs that we take, they are small chemical compounds, and understanding how those chemical compounds bind tightly with proteins is very important for drug discovery. So therefore, the question that was asked by experts, including our group and Google DeepMind and other top research group, immediately after the release of AlphaFold 2 and OpenFold, the question was, how can we extend the system ability such that on top of predicting proteins for a single chain or an individual protein, how can we predict protein-protein interaction, protein DNA, RNA interaction, so on and so forth? And the holy grail, something that I will not mention today, is really understanding the dynamic of those molecular systems. Something that I should have stressed, protein structures are important, but those molecular systems are not typically rigid. They are not Lego pieces. They have some fluctuations. Understanding their dynamics is the holy grail. And there is some important work along those lines, but I will leave it for another day. So we ask this question, how can we extend AlphaFold 2 abilities? And let's pick this example, protein-protein interaction. So we have a system that allows us to predict protein structure with high accuracy for single proteins. How can we extend it for multiple proteins? And what I want to stress here is really the importance of our system OpenFold vis-a-vis AlphaFold 2. So immediately after the release of AlphaFold 2, many academic groups, including Arne Elofsson in Sweden, they started kind of fine-tuning and tweaking with AlphaFold 2 to use the weights of the original system and make it such that it predicts like protein complexes. And they did very well. This is an example. This is, again, a metric, a benchmark. The higher, the better. The higher, the better means you have better prediction for protein complexes. And these are different variants of like this tweaking strategies, alpha linker, alpha fold gap, so on and so forth. And something just to notice is that those tweaking approaches, they were doing better than the best existing algorithm prior to AlphaFold 2, including ClosPro. So the situation was the following. We do a little bit of tweak to AlphaFold 2. We substantially increase our prediction abilities for protein complexes. But it turns out that the best system that we have today is, again, from Google DeepMind. It's called AlphaFold Multimer. And the main difference between AlphaFold Multimer and all the other versions is that AlphaFold Multimer was retrained from scratch, accounting for the inductive biases that correspond to the way how protein form complexes. So in a nutshell, the details of the protein-protein interaction problem are not very important. What is important to keep in mind, if we want to extend AlphaFold 2 and OpenFold, and if we want to achieve very high accuracy, we need to retrain the system from scratch. We need to include inductive biases that correspond to the problem that we are tackling, and that is the condition for moving forward. And as I mentioned earlier, the main advantage of OpenFold vis-a-vis AlphaFold 2 is that AlphaFold 2 version that is publicly available is not trainable. The OpenFold system, on the other hand, is trainable. You can use it, you can retrain it, you can add new modalities, and you can take it beyond the original AlphaFold 2. And I will later give you an example, again, coming from industry, from Meta AI, where they have used our OpenFold for kind of a new way of thinking about protein structure prediction. I will mention also a very important application that came after AlphaFold 2, which is protein design. So basically, we started predicting proteins that have been discovered in nature, that they exist in our organism. But a very active research area is to say designing proteins that were not discovered in nature, that do not exist in any organism. This, we call it Denovo design. And the strategies are very similar to the kind of strategies that you might be familiar with, diffusion systems like DALI for generating images. So this is an example from the Baker Group, a leading group in this space, where you start with target protein, and then you have a diffusion system that is designing a protein that can bind with your protein of interest. So this de novo protein design, combining AlphaFold 2, OpenFold with diffusion systems, is kind of opening major opportunities for drug discovery. Because basically, if you have a protein that you want to target and you believe it is involved in a given disease, those tools may allow you to design what we call a biologics, or like a drug that could inhibit this protein. So this is also a tremendous area of interest and research. So now let me turn a little bit into OpenFold in a nutshell, and again stress the extent to which OpenFold has advantages with respect to the original AlphaFold 2. As I said, OpenFold has been released now for more than a year. It is extensively used by researchers in academia and in industry. And again, quoting from Yann Lequin, it is open source, trainable, highly accurate, memory efficient, and GPU-friendly PyTorch reproduction of AlphaFold 2. I would have loved to tell you more about the architecture of AlphaFold 2 and OpenFold. Unfortunately, I don't think we have time. I will just mention a few vignettes. First, it is fair to say that this is, I believe, the most sophisticated machine learning ever built, not only in science. It's way more sophisticated than ChatGPT or DALI. I will stress a few things. So you might be familiar with the race today in LLMs and large language models, where you want to build larger kind of networks with more parameters and more data. AlphaFold 2 and OpenFold were trained using only 93 million parameters. So this is a drop in the bucket with respect to any system that we are familiar with in LLMs. And the reason is that we don't need as many parameters for training the system, because we have way more inductive biases into the system. Let me just pick one example. So first, AlphaFold 2 has the transformer architecture and the attention in its core. And that's why here you have this block called the evoformer, which is a combination between evolution and transformer, the evoformer. So the transformer is a key architecture in AlphaFold 2, but unlike ChatGPT and many other cases, the transformer by itself is not leading to highly accurate prediction. So let me, as I said, pick one example. In AlphaFold 2, there is the pair representation track, which is building representation over the pairwise distances between the atoms that make those proteins. But the pair representation is built in such sophisticated way, such that when the system is updating those pair representation, it tries to keep track of a very simple geometrical relationship, which is the triangular inequality. In a nutshell, we do know that if I give you any 3 points in space, in Euclidean space, they need to form a triangle, and the length of those edges that form the triangle, they need to fulfill the triangle inequality. A very simple mathematical property, but networks don't know their Euclidean geometry, so we need to do some sophistication to account for the triangular inequality. And if we don't do it properly, we see a major substantial drop in AlphaFold 2 performance. So that's kind of just a vignette. One thing also to notice is that even if the system is not using a large number of parameters, the system is very expensive in terms of scaling. The scale of the pair representation track is cubic, and this is a major limitation of the current AlphaFold 2. Anyway, it's a very sophisticated architecture. I cannot say much, but for those of you who are curious, I gave a series of lectures at the Harvard Center for Mathematical Science and Application, 3 lectures more precisely, where I really try to unpack for non-expert what is AlphaFold 2 and OpenFold, how they work, what is the underlying kind of inductive biases, and how we think about the transformer architecture. They're available on YouTube for those of you who are interested to learn more about those systems. So let me now briefly contrast AlphaFold 2 and OpenFold. So AlphaFold 2 is highly accurate. The predictions are, in many cases, achieving atomic accuracy. It has been built on JAX. Those of you playing with machine learning, they know that there are many platforms. JAX is the Google platform. The data for training AlphaFold 2 were not shared. They were not released. Those data are called multiple sequence alignment. So basically, it's taking advantage of the fact that proteins have evolved. OpenFold, on the other hand, is highly accurate, as I will show in a moment. We have used PyTorch instead of JAX, and that had like a major impact because, again, as many of you know, PyTorch so far is a way more popular platform for machine learning than JAX. We have released the data, and I will explain that in a moment. But maybe more importantly, and as I stressed earlier, the version that Google DeepMind shared with the rest of the world of AlphaFold 2 is not trainable. So you can only use it for inference. You cannot retrain it. Whereas OpenFold is fully complete. You can use it for inference, but you can also use it for training. And I will explain in a moment one important application of retraining OpenFold from scratch and generating fresh weights for the system. So I mentioned the data. So in a nutshell, much of the data that we use in this space, they are publicly available. We use structures, we use sequences, and they are available, but generating certain data is very expensive. And so we decided to share the data that we have used, in particular, the multiple sequence alignments and the templates, and the data are available. And we had like a NeurIPS paper discussing those data and how they are important. So I was insisting on the fact that having a trainable system is critical for exploring novel scientific questions. And the very first application of OpenFold came from Meta AI, or previously Facebook, where, in a nutshell, what they did, they have used a strategy which is very similar to ChatGPT that I will explain in a moment, and they combined this strategy with our OpenFold to predict protein structure. So the starting point is the following. You take your sequence of proteins. If you remember at the beginning, I said we are trying to find the map from a one-dimensional string of discrete characters, the amino acids, and we try to predict a three-dimensional object, a geometrical object. So you can think of those strings of amino acids as a text, like ChatGPT. You can use a masking strategy, like ChatGPT or BERT, and then you can build a system that is generating sort of embeddings for those tokens. So basically, the ESM2 system by Meta AI is the following. You take sequences from genomic databases that correspond to protein expressed in any organisms, from bacteria to humans. You use those masking strategy. You generate some embeddings. So this is the pre-trained stage. And then you take those pre-trained kind of representation and you feed them into our OpenFold. And because OpenFold is trainable, you can do that. You cannot use, again, the AlphaFold 2 system that is publicly available to train it using embeddings from a language system. So in a nutshell, what this ESM system is, it's a combination of a pre-trained masked language model, very similar to the techniques that we use for LLMs, and a folding pipeline that is coming from OpenFold. I have to say that on that front, we were a bit scooped, because our one of the main intention of building OpenFold was precisely to combine a pre-trained language model with our OpenFold. But as it turns out, Meta AI, they have more resources, more GPUs, so they went much faster than us. But we were scooped in a good way. So what this allowed the Meta AI team, it allowed them, by combining OpenFold with the protein language model, to predict the structure of over 700 million proteins from bacteria, viruses, and other organisms. So let me just remind you this idea about doing science at digital speed. Experimentally, we were able to characterize, until today, something slightly over 200,000 proteins. With AlphaFold 2, we went to 200 million proteins from 200,000. And with ESM and OpenFold, we went now over 700 million proteins. So we are entering a completely novel era in kind of structural biology. One thing maybe I should have mentioned earlier is that one advantage of those language model for proteins vis-a-vis AlphaFold 2 and the original OpenFold is that they are not as accurate as AlphaFold 2, but they are much faster at inference time. And that's the reason why Meta AI was able to fold 700 million proteins, way more than the DeepMind kind of system. On that front, combining protein language models and machine learning, deep learning for protein structure prediction, independently of Meta AI and prior to Meta AI, we had our own system that appeared in Nature Biotech, where basically we used a BERT-like strategy, and we combined the BERT-like strategy for a pre-trained protein language model with a sophisticated machine learning that is using ideas from mathematics and physics. And we were able to achieve better performance for an important class of proteins, such as orphan proteins and Denovo proteins. Now, OpenFold, I gave you one kind of application coming from Meta AI. And by the way, the team who did the Meta AI work now started a startup in the US called Evolutionary Scale. The system now is broadly available in like the major cloud services, such as NVIDIA and AWS. As some of you might know, NVIDIA and AWS are today interested in biology, drug discovery, and they are including OpenFold as a software into their platform. So now let me kind of share a few technical details about OpenFold. As I said, reproducing a code in itself is a major achievement, but the proof is really to achieve this very same performance of AlphaFold 2. That's kind of the major challenge, and we did it. So this is just an example. We have used Cameo, which is a well-established benchmark in protein structural biology. And what you see here on the y-axis, these are the predictions from OpenFold, and on the x-axis, the prediction from AlphaFold 2. Each dot here is a protein from the test set. And again, as you see, there is a very good match between the two. We achieve a mean of 0.91 LDDT, one being like a perfect prediction. So both AlphaFold and OpenFold are providing very highly accurate prediction for proteins. This is an example. Again, these are cartoon representations of proteins. What you see here in red or pink is OpenFold prediction, in blue AlphaFold 2, and in green the experimental kind of prediction. And as you notice, for just this example, there is a very good matching. And again, this is the statistical analysis showing that we have a very good match across the board. One important discovery we did and we achieved is the efficiency during training. As I said, we are maybe the only team, or among the very few, that have the ability of training systems like AlphaFold 2. And because we have optimized the system, we achieved something remarkable, which is that we can achieve 90% of the final accuracy of the system in only 3% of training time. And this is very important. So let me first unpack kind of this curve. This is a learning curve. Again, many of you might be familiar with those curves. The metric is LDDT, the higher the better, one being a perfect prediction. These are the steps during training. And if you see here in this region, what I was highlighting is that we achieve 90% of final accuracy very early. during training. So why this is important, in this observation suggests that we can train new OpenFold variants with far less compute. And this should allow us to rapidly explore new variants. And so this statement is very important because let me make kind of this observation. As you know, training LLMs is very expensive computationally. You need like thousands of GPUs, if not like more. We don't know the exact number for GPT-4, but it's a very large number. In our case, because we have optimized the system, we can train OpenFold using only 44 GPUs. But because we have this highly efficient training mode, in fact, we can achieve 90% of final accuracy using maybe a dozen of GPUs in less than a week. So the importance of that is that we can explore ideas very quickly and very efficiently, even if we don't have the resources of major industry players such as Google and Meta. Another important aspect about our system is that we are much faster than the original AlphaFold 2 during inference, and we can fold much larger proteins on an individual GPU than the original AlphaFold 2. And the reason, in a nutshell, as I said earlier, we have used PyTorch as an environment. We have used GPUs for training the system. So our kind of ecosystem is way more friendly than AlphaFold 2 for commercially available machines. So here, this is kind of just a table summarizing briefly on the first column the size of the protein. So those proteins, again, they have, as I said, tokens, amino acids. The number of amino acids is the length of the protein. And here we go from 100 to 4,000. These are the inference time in second. And as you see, OpenFold is faster than AlphaFold 2 on commercially available machines. And at some point, you cannot fold proteins on individual GPUs using AlphaFold 2. You are out of memory, whereas for us, we can go up to 4,000 residues. I have to say, this is a work that has been done in collaboration with Microsoft. They were really interested in our work, and they wanted kind of to help us to optimize certain aspects of AlphaFold 2. Maybe I should ask, do I still have some time for the slides, or is it the right time for asking questions? I would like to add a few things, but I also want to be mindful with your time, Abdelghani.
You still have time, yeah.
Okay, great. So now, great, thank you. I wanted to mention something very important, both conceptually, but also kind of with very profound practical application, which is the ability of the system to generalize and its scaling behavior. So again, some of you might have seen this plot, which is a sort of evolutionary tree for LLMs, right? Starting with GPT-1 and then GPT-2, GPT-3, all the way up to GPT-4 and now Llama and Falcon. I believe Marwan gave you a taste about the remarkable work he did with his team for Falcon. And so if really ask the question, how we understand this evolutionary tree and why we kept building stronger and more powerful systems based on LLMs from GPT-1 to GPT-4. And again, as many of you may know, an underlying idea is the scaling hypothesis. So there is this very important paper by OpenAI that was released 3 years ago in 2020 under the leadership of Jared Kaplan and Dario Amodei, and recently they started Anthropic AI, which is competing with OpenAI. And the most important thing of this paper is that they realized that the model performance is correlated with the amount of compute, the data size, and the parameters. So in a nutshell, what this paper is telling us, the bigger the better. If you want to achieve bigger performance, you need to increase your data size, your compute, and the number of parameters. And this has been sort of the guiding hypothesis among all the players today in LLMs. This kind of, from our perspective in science, and biology, drug discovery in particular, raises many questions. First, practical question. We cannot easily increase our data size. Generating data in science is way more expensive, and it requires a very sophisticated expertise, whereas I don't need to convince you, if you want to generate images and text, certainly on internet, you can take everything else. I mean, presumably much of the things on internet might not be of high quality, but still they are used for training LLMs. So we didn't, we were not able to do a systematic analysis in terms of like how OpenFold is scaling with respect to compute data and parameters, again, because it's very expensive and we don't have the resources of OpenAI or Google. But we started asking the questions about data because of the critical role that data play in scientific kind of exploration. So let me unpack a little bit this plot that is kind of leading to a profound, I believe, discovery and observation. So again, what you see here, these are different like training curves. Again, we use the same metric, LDDT, the higher the better, one being perfect prediction. And on the x-axis, I'm plotting the training steps. So we start training the system using 130,000 proteins. So these are the experimentally available proteins on the protein data bank. This is how AlphaFold 2 was trained. This is how OpenFold was trained. First observation, this number in itself is not that high, right? If you have played with images and text, to train a CNN for images that is very sophisticated, you need millions of images. So we don't need that many data. But then we ask the question, imagine we don't have as many data as the protein data bank, and we started like randomly reducing the number of data for training the system. So next, we decided to train the system using 15,000 proteins. And what you notice here is that we achieve exactly the same performance as the original system using the whole data set. So we kept decreasing the amount of data. We picked 10,000 proteins. This is this curve. And again, we achieved the same performance. We went to 5,000 proteins. There was a drop in the performance, but not dramatic, not drastic. And then we went all the way down to 1,000 protein, which is the blue curve here. There is a drop in performance. However, we believe that our blue curve is still better than much, if not all, the systems that were released before AlphaFold 2. So what is the moral of this story? So the moral is that we may not need as many data as what is expected from what we have learned by training ChatGPT and DALI and other systems. The scaling behavior might be very different. We don't see at least a clear scaling between performance and data size. And as I showed you, training the system with 130,000 proteins or 10,000 proteins is leading roughly to the same performance. So this is extremely important because I believe we might have discovered a regime that is different than the regime described by LLMs and ChatGPT. It is also very important because it might provide optimism in the following sense. We don't have many data in science, including biology and drug discovery. We don't expect to substantially increase the amount of data, or at least certain class of data, in the coming years. And so this analysis suggests that we can still achieve very high performance with the data that we have today. And if you ask the question, why then is it the case that AlphaFold 2 and OpenFold have a very different behavior than, say, ChatGPT? Here I can only speculate, but my speculation is the following. Language, human language, is the outcome of centuries of contingent evolution that includes history, politics, culture, whereas biological systems and scientific systems are highly constrained by the underlying physical, chemical rules. So therefore, the space of solution for proteins, but also beyond proteins, is highly constrained. And therefore, if we build a sophisticated AI architecture that account for the inductive biases, the right inductive biases of those systems, we can achieve very high accuracy without using that many data. And so let me highlight why this problem is really critical for us and the experts in the field. So this is like data sparsity in biology. And so what you see here on the left, this is a curve that is reporting the amount of proteins for which we have a structure experimentally in the PDB. So we have something around 600,000 chains in the PDB, and like we do some filtering and curation, but these are like the raw data that we use for training AlphaFold 2 and OpenFold for protein structure prediction. What you see here on the right plot, this is the amount of RNA structure that are available experimentally. And as you see, there is very little. We have only 9,000 chains that are available in the PDB. And there is no hope in the foreseeable future to experimentally increase the amount of RNA that are available in the PDB. And I believe I don't need to tell you much why RNA is important. It is important to understand biology, but today it is becoming way more important for therapeutics. One example is the COVID vaccine from Moderna and BioNTech and Pfizer that is based on the RNA technology. So understanding much better RNA structure, RNA function is a critical piece, and we don't have as much data. And so I believe the only challenge that we are left with, at least for the near future, is to build very sophisticated machine learning algorithms that account for the inductive biases with the hope of achieving a highly accurate system with RNA. We don't yet have this system. I am working in collaboration with Dr. Elena Rivas, my colleague at Harvard and a world expert in RNA biology, and we are trying to we are trying to build a novel OpenFold for RNA structure prediction. So very briefly, next steps. I mentioned earlier that predicting protein interaction with small molecules that you see here or drug-like molecules is very important for drug discovery. This is a major question for pharma, and we started taking this journey. So in a nutshell, we are currently training a system that we call OpenBind, where now as input, we take those sequences of amino acids that I mentioned earlier, a discrete one-dimensional string of tokens. But on top of that, we take two-dimensional molecular graph that correspond to chemical compounds. And the system now, instead of just folding proteins, it is co-folding proteins with small molecules ligands. So we are very excited by this work, and I hope I will be soon able to kind of update you, or like we will certainly share our system and weights. The results that we see now are very encouraging. So let me maybe conclude with that and say, where are we going? The most important thing from my perspective with AlphaFold 2 and OpenFold, it's not really the ability of predicting protein structures, which is in itself a major question in biology. It is really telling us that we might be now be able to make predictions in biology and drug discovery. So if you have physicists in the audience, you do know that a major contrast between physical sciences and engineering on one hand and biology and drug discovery on the other hand is that physics is based on mathematical modeling, simulation, and predictions, and that's why we are very successful on that front, even from an engineering perspective. On the biology side and the drug discovery side, unfortunately, we don't yet have predictive tools. So we turn to experiments and we use a sort of a craft, and that's why we are not able to cure diseases such as cancer and neurodegenerative diseases, just to mention few. What AlphaFold 2 is telling us, and OpenFold, we might be able now to systematically have highly accurate, efficient, and scalable predictions and simulation for biology. Protein structure prediction is the beginning. I gave you an example, protein small molecule interaction, but RNA structure prediction, protein-protein interaction. And collectively, this may allow us to have a way more predictive biology and drug discovery. And this could be the beginning of a completely new way of dealing with biology and drug discovery. I will conclude by mentioning the key elements of the OpenFold team, the different organizations that were allowing us to train the system, including NVIDIA, TAC, and DARPA. Thank you very much for your time. I hope I was able to convey part of the excitement and part of the breakthroughs that are happening in our space. Thank you.